for the delay. We've had some very serious technical hitches, and our two chairs who are supposed to be running the session have not been able to log in for some reason, and I've been drafted in at the last minute, and I apologize, I haven't actually have any preparation for our two wonderful speakers today. But we've got two speakers. We've got June Round, who's going to give us a talk about adaptive immunity and beneficial interactions between the host and fungi. And then we've got Damian Krysan, who's going to be talking to us about Candida albicans filamentation in vivo. I'm not going to do the introductory slides today because we've already lost 8 minutes of our allocated talk time. And so with that, June, thank you very much. And apologies again for keeping you hanging while we have this technical glitch, and I will introduce June, and we look forward to your presentation.
Thank you, Gordon. I appreciate it. So I'll go ahead and share my slides. Oops. Then is that working?
Yep, yeah, and you're in presenter mode. It's all perfect. Off you go, June. Thank you.
Great, thank you. Well, hello, everyone. Good morning or good afternoon, depending on where you are. As Gordon said, my name is June Round. I've been at the University of Utah for the last 13 years. And as you'll see from the title of my talk, I decided to at the last minute to change what I was going to present to you today, because I noted that about a year ago, a very talented postdoc that came out of my lab, Kyla Ost, presented on MycoTalks, and she talked about the project that I had originally planned to talk to you about. So I thought instead of re-going over that published data, that I would share a newer story that is going to start with bacteria, but I promise in about 7 slides will bring us back to the fungal community. So just a little bit of background about my laboratory. We're interested in the interaction between the commensal microbiota and the immune system. So I'm a trained immunologist who kind of fell in love with microbes during my postdoc. And we're interested in how this relationship influences or promotes health. So we view relationships with microbes a little differently than most, in that we believe that there are many, many microbes that are really instrumental in many processes that are important for development and prevention of disease. So oftentimes we are looking for organisms not that are present during the disease state, but rather are present during the healthy state and perhaps lost during disease. So we study this relationship as it pertains to many diseases such as inflammatory bowel disease, multiple sclerosis, but today we'll be talking about a project that is involved in type 1 diabetes. So I often have to remind the groups that I'm talking to that the microbiota is a very complex community. Most of the studies in this field tend to focus on the bacterial component, but we're also home to archaeal species, many, many viruses, not just eukaryotic viruses, but also prokaryotic viruses, of which a section of my lab also studies their interaction with immune system. But as this community likely appreciates, we're also home to a vast fungal community. So we'll be talking about some of those members today. So this is a project that represents a collaboration with some really wonderful individuals. All the work that I'll be talking about today is performed by another great postdoc that I've been able to interact with, Jennifer Hill. She'll be starting her own lab this summer at University of Colorado Boulder. So this is her work I'll present to you today. She did her PhD studies in the laboratory of Karen Gilliman, who is a collaborator on this project, working on zebrafish and pancreatic beta cells, which we'll talk about today. And then I also collaborate on this project with Charlie Murtaugh, who's at University of Utah, and he is an expert in pancreatic development. So this has been a really fun project to work on with these really talented individuals. So the focus of the talk today will be honed in on these pancreatic beta cells, which I'll be honest, I didn't really think much about until Jennifer Hill joined my lab. But as many of you know, these beta cells are really important for our metabolic regulation in that they are the producers of insulin, and they help us to regulate our glucose levels. So these pancreatic beta cells are the targets of destructive autoimmunity, in that T cells will infiltrate into the pancreas and they'll start to kill off these beta cells. And of course, when you kill off cells that produce insulin, you no longer produce insulin, and you can no longer regulate your glucose levels. So of course, one of the major treatments for type 1 diabetes then is going to be exogenous insulin production. And this has really been the standard of care treatment for type 1 diabetes for over 100 years. So new therapies, new preventatives are really needed for this particular disease. So when thinking about an effective therapy or an effective cure, it's really going to require two components. The first is going to be blocking that autoimmune infiltration into the pancreas and destroying those beta cells. So that will really have to be one of the first things that happens. But then, of course, once beta cells are destroyed, they really don't naturally overturn. So once they're destroyed, you don't get them back. So another component of an effective therapy is going to be the ability to regenerate or replenish lost beta cells. And really, this is going to be the implication for some of the work that I'll share with you today. So as we develop, of course, we acquire our microbiota, but also this early life component is very important for the development of type 1 diabetes. So in thinking about the genetic elements of type 1 diabetes, over 70% of individuals who go on to develop type 1 diabetes have this predisposing MHC or HLA risk allele. But interestingly, only 3 to 7% of children with this particular allele will go on to develop type 1 diabetes. And thus, by no means is having this allele a predictive factor for the development of type 1 diabetes. So it's been appreciated for a long time now that there are a lot of environmental factors that are important for the development of type 1 diabetes. One of these factors that has been investigated for some time now, of course, is the microbiota. So here I'm showing you a piece of data from this paper from Ramnick Xavier's lab, and they followed the microbiota of children that possessed this HLA risk allele. So they had the predisposing genetic element for the development of type 1 diabetes, and they followed their microbiota from the time they were born to the time that they're 3 years old. Many of these individuals were diagnosed with type 1 diabetes right before they turned 3. And when they look at the development of type 1 diabetes and compare the microbiota in these individuals, they find that individuals that go on to develop type 1 diabetes have a significantly reduced microbiota when compared to those individuals that do not. So this data has always been intriguing to me because this really goes along with the idea that perhaps disease develops because you're missing important microbes that are important for promoting the healthy state. So this really suggests that there is some microbes that might be missing that prevent type 1 diabetes development. So an important element to know about these pancreatic beta cells is that they really undergo an important proliferative birth really early on in life. Around the ages of 1 and 2, these beta cells, you're born with a certain level of beta cell mass, and then as the infant grows, to keep up with the growing mass, these beta cells undergo a huge expansion event. And it really sets essentially the number of beta cells that you'll have for the rest of your life. And again, beta cells don't replenish very well over time. So this developmental window is really important for producing the beta cells that you'll have for the rest of your life. And coincidentally, it also coincides with a really important time frame where you're acquiring and diversifying your microbiota. So these two events are occurring at the same time. So when Jennifer Hill joined my lab, she had done some work in zebrafish to show that the microbiota in zebrafish was really important in instructing the amount of beta cells in an adult zebrafish animal. And we had collaborated with Karen Gilliman's lab at the time to show that this was also true in a mammalian mouse model. So here I'm showing you data that's, we'll be looking at beta cell mass for the rest of the talk. So this is basically shows you how many beta cells in your insulin producing capacity. And in SPF animals, which are your normal colonized animals, compared to animals that don't possess a microbiota or germ-free animals, we can see that germ-free animals have significantly reduced beta cell mass. And this is true in every background that we've looked at, and I'm showing you two different backgrounds here. And these are just adult animals. So this suggests, again, in the mammalian model, that indeed the microbiota is important for kind of setting the amount of beta cells that you'll have in an adult. So I just told you how important this neonatal window is for the development of beta cells. So when Jennifer Hill joined my lab, she wanted to ask questions about, well, is the microbiota important during this early life window, and are there specific members that are involved? So she set up this really interesting experiment where she disrupted the microbiota using what we call an antibiotic bomb. So it's 4 different antibiotics: metronidazole, vancomycin, ampicillin, and gentamicin, I believe. And she provides these antibiotics over a 10-day window and then withdraws the antibiotics and then looks at animals in their adult state. So just to describe these windows, there's 3 of them. The first window is providing antibiotics while animals are in embryonic day 14 until postnatal day 4. The antibiotics are then withdrawn, and again, the animals are allowed to grow up, reacquire a complex microbial community, and they're analyzed at adulthood. Similarly, there's a window where antibiotics are provided between postnatal day 4 and postnatal day 12. Again, antibiotics are withdrawn. Animals are allowed to grow up to adulthood. And the third neonatal window is P12 to P21. So when we pulse these mice with antibiotics, which might be akin to what children would receive if they have an ear infection. I know many of my children received antibiotics early in life. When we go and we look at animals that receive these antibiotic pulses for beta cell mass, we found something pretty interesting. So just to orient you, we're looking at beta cell mass in SPF and germ-free animals. I already showed you this data in the slide before. So germ-free animals have significantly reduced beta cell mass. If we look at animals that received antibiotics throughout their life, they look very similar to germ-free mice. And if animals received just 10 days of antibiotics in embryonic day 14 to P4, or in that second pulse window, which is postnatal day 4 to P12, they look very similar to SPF mice. In fact, there seems to be no perturbation to their beta cell mass. However, strikingly, when animals receive just 10 days of antibiotics between postnatal day 12 and postnatal day 21, this third pulse, if you will, we see a very dramatic influence on beta cell mass, in that just receiving those antibiotics during those 10 days makes those animals have the beta cell mass similar to a lifelong germ-free animal. So this suggests that this 10-day window of development in these mammals is really important for the microbiota effect on beta cell development. So this is just not on, I'm going to see if I can actually put my pointer on, maybe that will be easier. So this is not just important for the beta cell mass, but it's also functionally relevant. So if you look at blood glucose levels in these, what we'll now term neonatal antibiotic treated mice, these are mice that we're looking at that only received antibiotics during this third window, and then again looking at them in adults. Animals that receive this pulse 3 of neonatal antibiotics have significantly higher fasting blood glucose levels. If they're provided glucose, animals that received these neonatal antibiotics are unable to clear glucose as quickly. And these are just animals on a normal chow diet. We haven't put them on a high-fat diet. And based off of these results, it's probably not surprising that if you looked at the insulin levels in these animals animals that receive neonatal antibiotics have significantly reduced insulin production. And again, if you give them glucose and look at the amount of insulin produced in response to glucose, animals that receive neonatal antibiotics do not produce as much insulin in response to a glucose challenge. So all of this data really points to an important developmental window of time whereby the microbiota is really important for instructing beta cell development. So of course, one of our questions was what microbes are relevant during this particular period of time. So this is going to start by looking at bacterial organisms, because we did start this experiment with antibiotics, and all of these antibiotics are known to perturb bacterial communities. So this is a 16S, and here we're just looking at the differences in bacteria between this window starting or opening at postnatal day 12 to P20. So this is between the window of time that we showed to be important for beta cell development. And each color essentially represents a different family of bacteria. And you can see that if you look at P12 to P20, there is a pretty significant increase in the diversity of organisms that are acquired in these animals over that window of time. So it's a really important diversification of the microbial community. So in order to identify organisms that were involved in beta cell development, we took an approach where we treated animals over that window of time with single antibiotics, which are known to perturb microbial communities in different ways. And again, I'm just showing you 16S data to demonstrate that when, for instance, when you treat an animal with ampicillin and look at what happens to their microbial community, it looks very different than when you treat an animal with moxilactam, where you see an expansion of, Streptococcus and other bacilli. So in this way, we can ask, well, what happens when you perturb the microbial community in different ways? So when we do this experiment, and look at beta cell mass, again, just to orient you to what beta cell mass looks like in a normal animal versus a germ-free animal, you can see that when you treat with antibiotics such as ampicillin, cephalothin, or chloramphenicol, that these animals have a beta cell mass that looks very much like a germ-free animal, whereas animals that were treated with moxilactam, for instance, they have a beta cell mass that looks like an SPF animal. So for these animals here that were treated with these single antibiotics, they either lost an organism that was important for promoting beta cell mass, or perhaps in moxilactam animals, they were allowed for expansion of an organism that positively influenced beta cell mass. So in this way, we're able to ask, well, what changes in the community occur, and what organisms can we follow up? So when we do an ANCOM analysis focusing in on these bacteria, and we'll talk about the fungi in just 1 minute here, in animals where you have a very high beta cell mass, basically to SPF levels, we see an enrichment of organisms such as Enterococcus, Lactobacillus, and Streptococcus. Whereas in animals that had a reduced beta cell mass, such as ampicillin-treated animals, we saw an enrichment of specific Bacteroides species, as well as E. coli species. So because at the time we had several people in the lab working on different fungi, and we know that when you antibiotic treat, that you can often open up niches where fungi can be enriched, or in instances where fungi and bacteria have a relationship, maybe getting rid of a bacteria can lead to a loss of particular fungi. We wanted to also look at the fungal community in this particular window. Now, generally in my facilities here at the University of Utah, when we look at the fungal community in our adult animals, we have very little fungi in our adult animals, and very little fungal diversity. But when we looked in our animals over this window of time, so when they're infants, and we focused in largely on Candida, because we know that Candida has been reported in many animal models as being present in infant animals. we saw that in infancy, there was this very large bloom of Candida, different Candida species at P12 that significantly dropped by the end of our developmental window, P20. Moreover, if you look at different fungi in these different antibiotic treated groups, we see that there's different levels of fungi present when you perturb even the bacterial community. So this made us interested to look at bacterial and fungal members and how they might influence beta cell mass. So what Jennifer went on and did at that time is she started to isolate organisms from a neonatal animal, and she isolated several Enterococcus species, some Lactobacillus species, and then she pulled out some fungal organisms from humans and mice. So here we have a Candida isolate, and then we also used Candida albicans, just the very typical Candida albicans used in most studies. And what she did is she took these organisms and she monoassociated germ-free mice, and then she analyzed the beta cell mass in these monoassociated conditions to see which specific organisms were really important. So again, germ-free levels of beta cell mass, SPF levels are significantly higher when you monoassociate with an Enterococcus species, two different ones that we isolated from mice, or this particular Candida species. We see that these organisms alone are able to induce beta cell mass to SPF levels. However, if you colonize with the Lactobacillus species or Candida albicans, we see that these organisms are unable to influence or induce beta cell development in these mice. So we found this very surprising, that Candida albicans, but this particular Candida species, which is closely related to Candida albicans, wasn't able to influence beta cell mass, but this particular one was. So this suggests that there are specific bacteria as well as fungal organisms that are important during this very narrow window of developmental time that can positively influence beta cell mass. So we were interested in, of course, the mechanisms by which this occurred. And during her PhD studies in zebrafish, Jennifer was really focused in on direct mechanisms by which bacteria could influence beta cells. And she published two really amazing papers, most recently one in Cell Metabolism, where they identified a mechanism that they called BEF-A, which is essentially like an antimicrobial peptide that penetrates the membranes of these beta cells and causes their proliferation. Again, this was all work done in zebrafish. So we had done a few experiments in the lab with mice, and we couldn't find any evidence that the organisms that we were working with were directly influencing beta cells in this way that they had found in zebrafish. So we started to focus in on more indirect mechanisms. And since we're more of an immunology lab, we were really interested in the immune-mediated mechanisms that might influence beta cells. And we had done an RNA-seq, and for the sake of time, I'm not going to show you that data, but we had done an RNA-seq in islets isolated from germ-free and SPF mice to understand what the major differences were. And interestingly, what we found is that germ-free animals had significantly reduced transcripts for one of the only immune cell types found in the pancreas, and that was macrophages. So germ-free mice lacked a lot of the different macrophage transcripts that you found in an islet of an SPF animal. So this got us really interested in trying to understand, well, how does the microbiota influence these macrophages? And in the literature, it is well known that macrophages are also really important to developing beta cells, although the mechanisms by which this happens are still quite unknown. So the first question that she wanted to ask was, are these neonatal macrophages, do they play any role in beta cell function and long-lasting metabolic capacity? So one of the first things that she did was to look at over this same window of time that we've shown the microbiota is important for instructing beta cell development, are these macrophages also important? So the way she approached this was multiple ways. I'm showing you the data for one, is she used claudrinate liposomes, and these are known to deplete macrophage populations. So she provides these liposomes over this 10-day window, and then she stops injecting the liposomes, macrophages rebound, and then she analyzes beta cell mass in the adult animal. So when she looks at the amount of insulin-producing tissue in the pancreas of these animals, she sees that when they have had neonatal depletion of macrophages, that adult animals have significantly less insulin-producing tissue or significantly fewer beta cells. Additionally, these animals produce less serum insulin when they've had neonatal depletion of macrophages. And again, if you provide animals that had neonatal depletion of macrophages, they're unable to clear glucose as quickly as animals that possessed macrophages during that developmental window. So we did this in addition in different ways of depleting macrophages, because we're aware that claudrinate can have off-target effects. So we used DTR-mediated ways to deplete macrophages, and we found the same phenotype. So again, this suggests that macrophage function is really important over this same time window in order for instructing beta cell development. So the last question that we wanted to ask, and I think my time's coming to an end, so I only have one or two more slides. We wanted to ask whether or not these specific bacteria and fungi that we had identified that were important for promoting this beta cell effect, are they acting through macrophages? Because again, we found no evidence for direct mechanisms of beta cell development. So the first thing that I'll show you is the influence of these specific organisms on the macrophages themselves. So interestingly, macrophages in the pancreatic islet are also dependent on the microbiota. This was perhaps a little less surprising to us because they're important for development of so many immune populations. So here, macrophages within the islet between an SPF and a germ-free animal, significantly reduced in a germ-free animal. If you take these germ-free animals and you monoassociate them with these organisms that I just showed you are important for instructing beta cell mass, we find that most of these organisms in a monoassociated state are able to induce macrophages to a certain level in the islet. But really interestingly, this particular Candida isolate is really good at inducing macrophages. So this was probably one of the best inducers of macrophages within the islet. So because of this, Jennifer wanted to test whether or not any of these organisms relied on macrophages in order to have their beta cell promoting effect. So again, she took two monoassociating germ-free mice and here we're focusing on two organisms, one of these Enterococcus species, and then our Candida isolate. And then she depletes the macrophages again with claudrinate over this window of time, and then analyzes beta cell development. And when she does this, she found that when she does this in the setting where animals are monoassociated with the Enterococcus species, that depletion of macrophages doesn't seem to affect the ability of this organism to promote beta cell mass. So this suggests that there are other mechanisms by which Enterococcus does this. However, when she looked at animals that were monoassociated with this particular fungal isolate, this Candida species, and depletes macrophages, she now sees that this Candida species is no longer able to have this beta cell promoting effect. And therefore, it appears that this fungal species is acting through macrophages in order to promote beta cells during this very important developmental window. So just to summarize, we've identified that there is a very narrow window of developmental time in mammals in which disrupting the microbial, bacterial, and fungal community is really important for metabolic health, in that even just these small perturbations early in life can have lasting consequences to the development of the animal. And here we've identified a novel fungal isolate that seems to have really important activity in inducing macrophages and then promoting metabolic health later in life. So I started this talk focused on type 1 diabetes, and can we use this perhaps to improve this disease? So I'll leave you with this last data slide, which is where we took NOD mice. So these are non-obese diabetic mice. They are germ-free animals. And here, these animals will spontaneously develop diabetes. And in germ-free animals, in our colony, we get the same incidence of diabetes in our germ-free animals as we do the SPF. So we're using the NOD mice to ask questions about, well, how do specific organisms impact the incidence of diabetes? So here we monoassociated animals with a lactobacillus species, which I just showed you data does not have any beta cell promoting activity. And then we also monoassociated another group of NOD animals with that fungal isolate, this Candida species, which does have very potent macrophage activity and beta cell promoting activity. And interestingly, in our initial experiments, we find that animals that are associated with a non-beta cell promoting bacteria go on to develop a lot of diabetes. In fact, 80% of animals develop diabetes, whereas animals that have this beta cell promoting fungal isolate do not develop diabetes to the same level. So this is perhaps just a preliminary piece of data to suggest that there are organisms present during that neonatal window that can be important for directing beta cell development and prevention of diabetes. So we're continuing to do that work. So again, I've tried to highlight the important people as I went through this talk. Jennifer Hill, again, did all of this work. I also want to highlight my lab manager, Ricky Bell, who does all of our germ-free experiments, and these NOD mice are really difficult. I have just a fantastic group of people that I get to work with, including many wonderful collaborators, which I've already highlighted. So I will stop and allow Damian to share his story. Thank you.
Yeah, thank you very much, June. Good news is that your chairs have also arrived by now.
Great.
Sorry for the technical issues, but obviously Gordon was so keen on sharing your talk that he did not admit us into the meeting. So as you know, we will do the Q&A after the two talks. So please, everyone, send your questions and also indicate that these are questions for June or Damian. And I will now hand over to Ilian to introduce our second speaker.
Thanks, Oliver. And I'll start with my introduction. So I'm glad to have here Damian Krysan. So Damian is a physician scientist at University of Iowa College of Medicine, and he's also the division chief of pediatric infectious diseases. As well, he has a chair in pulmonology, allergy, and infectious diseases. So Damian's career has gone in a very interesting way. So from chemistry to biological research in a company. After that, he did his MD, and right now he's a physician scientist. So his PhD is in organic chemistry for Northwestern University. He did his postdoctoral training in Emory University, and he spent 5 years basically in Abbott. And he was working on HIV protease inhibitor, which is on the market for a long time now, ritonavir. So he returned to medical school after that and received his MD at Michigan. Then he spent 12 years in University of Rochester as an independent PI and moved to Iowa in 2017. So his research focuses on two major fungal pathogens, Candida albicans and Cryptococcus neoformans, where he explores mechanisms of virulence and commensalism. In addition to that, he has applied his training in organic chemistry, and he's working on some very interesting screens and preclinical developments in new antifungal molecules. So today he will talk about basically the talk is moving inside. It will be focused on Candida filamentation Damian, looking forward to your talk.
All right, thank you for that nice introduction. And let me get my slides up here. All right, go to the top and not the bottom. There we go. All right, so thanks again for the opportunity to participate in the MycoTalks. Today I'll be telling you about a story we've been working on for a long time on looking at how Candida albicans actually filaments in an infection model of candidiasis. And so if you've been in Candida or fungal pathogenesis for any length of time, you know that lots and lots of folks have worked on Candida albicans filamentation over the years. And the reasons for that are that the ability of Candida to undergo this switch from a round sort of budding yeast form to the long and skinny hyphal filamentous form plays important roles in multiple steps of pathogenesis. The yeast form, as we've learned over the last few years now, it promotes GI colonization. It's sort of necessary for dissemination in the bloodstream, but the yeast-locked cells, so to speak, appear to cause very little or no tissue damage in mouse models. In contrast, filamentous form is less fit in the setting of GI colonization. In general, they don't disseminate very well because they're presumably big and stick in various parts of the bloodstream and don't get to their target organs. But once they're in the target organ, the hyphae is critical for causing tissue damage. And we look in the literature, which I did about 6 or 7 months ago, and you do a search term of Candida hyphae, and you probe the PubMed database, you end up getting about 2,500 papers have been published on this yeast to filament switch in Candida albicans, which is quite a bit. If you look at the Candida genome database, there are over 240 genes now that have been characterized as having decreased filamentous growth. And this might make you wonder how much more do we need to know about this process? And my take on that is that most of this has been done through studies involving in vitro induction of this morphogenesis change. And my son happens to be a philosophy major, and I came across this looking through actually one of his books. And it's a quote from Werner Heisenberg, who's famous for the Heisenberg uncertainty principle. And he said a number of years ago, we have to remember that what we observe is not nature itself, but nature exposed by our method of questioning. And so with this sort of notion, I was interested in asking whether, the method of questioning affects particularly in vitro Candida albicans filamentation. And this is some data that from a different project focusing on in vitro filamentation, in which we have a protein kinase knockout, which when we look at its filamentation phenotype, which is this fuzzy stuff around the outside of the wild type strain, on spider media, when we knock it out, it goes to a very smooth colony form indicative of a reduction in the ability to undergo filamentation. So if we took that media, we'd say BCK1 is absolutely required for filamentation in Candida albicans. However, if we switch to RPMI plus 10% serum, as you can see, we have these radiant invasive filaments coming out from the center of the colony in both the wild type and the BCK1 mutant. And so in this filamentation phenotype, conditions really matter. And this has been shown, I think, with increasing regularity over the last maybe 10 or 15 years, that Candida filaments in different ways depending on the environment in which it finds itself. And so our hypothesis for this work was that if the method of our questioning matters, to go along with Heisenberg, then studying filamentation in infected tissue might provide new insights into this really important Candida albicans virulence trait. And so this is what we've been working on now for a number of years now. And the model in which we have developed to study in vivo filamentation is as follows. We take advantage of the fact that the mouse ear is very translucent. And we inject fluorescently labeled Candida into the subdermal stroma beneath the sort of keratinized layer of skin. And we can actually observe these cells in real time undergoing filamentation with confocal microscopy. And what we're modeling here is basically the filamentation of Candida in a subepithelial anatomical compartment. Now, Candida is a colonizer of mainly oral and GI mucosa, which is another type of epithelium. But when it undergoes translocation as part of the initial steps of disease formation, it transitions through this epithelium into a subepithelial stroma. And that is exactly where we are observing this morphological transition when we use our model. In disease states such as oropharyngeal candidiasis, this is where it stops. And the disease is really driven by the hyperformation within this stroma beneath the epithelium. In the setting of disseminated disease, the sort of standard model now is that once they're invaded through the epithelium, the Candida gain access to the bloodstream, and if the patient is lacking neutrophils, for instance, it disseminates onto an organ. And so that's sort of the premise behind our use of this somewhat unusual way of infection to directly study the morphological transition. We also take cells harvested from the ears or directly in the ear tissue and do in vivo profiling of the Candida albicans gene expression during this filamentation process. So those are the two assays that we're really going to focus on for the rest of the talk. And the first question you might be asking is, well, how does filamentation in the ear compare to filamentation in more standard in vitro conditions? And so we asked the same thing and did an experiment where we directly compared in vitro with RPMI and 10% serum at 37 degrees, compare that over different time points to filamentation within the ear. And if you look in vitro, you see about 40% of the cells have formed filaments within the first hour. By 2 hours, they have peaked at 80–85%. And then there's actually a slight but statistically significant decrease in the percentage of filamentous cells as you go out to 8 to 12 hours. When we look in vivo, we see almost immediate, as it were, within the time course that we're looking at, within an hour, we see a very strong filamentation. And that filamentation, pretty much the percentage doesn't change over the next 12 hours. In contrast to the in vitro conditions. As you might imagine, we can also directly measure filament length in this assay, and the filament lengths slowly increase to a maximum and then kind of stay at this peak through the rest of the time course. And the similar pattern is seen in vivo. So the main difference here is that takes a little longer for in vitro to get going, and then it appears to taper off. Whereas in vivo, we don't see any real change in the total percentage of filaments that are present over at least 12, and we've done this out to 24, and it really doesn't change at 24 hours either. So the next thing we looked at is the expression of some of our favorite hypha-associated genes in this time course, both in vitro and in vivo. And the in vitro data are shown in black and the in vivo data are shown in red. And as you can see here, there's in vitro in particular, these genes are expressed at higher levels, at absolute levels, relative to the in vivo per, microliter of sample as we're using here. But there's a big peak in vitro, and then there's this fall off. Whereas in vivo, we see a relatively slow increase in expression, but it maintains for these transcription factors, UME6, BRG1, TEC1, and for the hype-associated downstream targets, things like ECE1, HWP1, ALS3, we see a plateau that is basically maintained over the time course of filamentation, indicating that this is consistent with the morphological data in which we see the in vitro cells reaching a peak and then starting to decline in the percentage of filamentation. So this program goes on and then goes off under this time course in vitro. It goes on and pretty much stays on in vivo. We also looked at, we're using nanostring here for this, these assays, and we compared the total expression profile using 186 genes, which are environmentally responsive as well as hyper-associated. And if we compare expression of the in vivo in the ear to that of the RPMI plus serum, we get a reasonable correlation between in vivo and in vitro. Clearly, there are differences in extent for many of these genes, and the correlation is pretty good. Similarly, when we look at kidney samples, we see a very similar correlation. But the best correlation comes when we look at the expression of these genes in the two in vivo settings, in the ear and in the kidney. And so to a first approximation, what's going on in the ear, transcriptionally at least, looks like it's very similar to what's going on in our favorite target organ in the mouse model, the kidney. But if you look at some particular genes, you see things that are expressed at, say, 1,000 normalized count here in the kidney, but they're more like 100,000 in the ear. So, like many things, there are similarities and differences between these two niches. And so the next thing we did was to ask what transcription factors then govern this process in vitro, and are they the same transcription factors that have been characterized as having effects, important effects in vitro. And what we did was we used our ear model and screened the Holman collection of transcription factor deletion mutants. And this work was published just about a year ago and was done, a heroic piece of work by my postdoc Rohan Waqaid. And what we found was that we had three of our fairly familiar regulators, BRG1, ROB1, EFG1, clearly stood out as having the largest effect on the initiation of filamentation in vivo. These data were collected after 24 hours of infection. And then we had some other ones that we call auxiliary regulators, which were statistically significant, but were not quite at the same level as BRG1, ROB1, and EFG1. We only found 2 regulators that increased the percentage of filamentous cells, and those were 2 well-known repressors of filamentation from in vitro studies, TUP1 and NRG1. And when we look at this data overall, what we found that there were 13 regulators that were in common between in vitro and in vivo, but there were 9 that were only appeared to regulate filamentation under in vitro conditions, and 6 that were had effect in vivo, but not under any in vitro conditions that we could see in the literature. So the relationship between the auxiliary and core regulators that we propose, at least to explain some of this data, is that the auxiliary regulators, when we look at them, typically we're responding to specific environmental conditions in the host. Things like iron starvation with CEP1, pH with RIM 101, hypoxia with CPH2. And these ones didn't have quite the same effect as the core, but these were sort of modulators in our model. And they work together with these core regulators to establish a sort of wild type levels of filamentation. If you lose some of these inputs, you reduce the filamentation a little bit, but if you lose the core regulators, it goes down a great deal. And if you're interested in more of this, you can, it's published data in our eLife paper. The next thing we looked at was a question about, do the regulatory pathways themselves function similarly in vitro and in vivo? With this, it's about a 10-year-old figure from Pete Sudbury's review. We had just looked at the bottom end of this, and now we're going to think about what's going on at the top. And the pathway we picked to interrogate was the cyclic AMP protein kinase A pathway, which is kind of a central regulator absolutely required for in vitro filamentation. And so this is where things got kind of interesting. So the cyclic AMP pathway is thought to respond to temperature, serum, glycopeptides, CO2, a whole bunch of different in vitro inducing conditions to generate cyclic AMP, which then activates the protein kinase module, which then phosphorylates EFG1, and through a less well-known or well-characterized mechanism, inhibits the function of NRG1 and TUP1, which are repressors of filamentation, and then we have our filament. And so we asked how well does this pathway correlate between in vitro and in vivo? Well, at the bottom of it, EFG1 is absolutely required for in vivo filamentation. If we knock out NRG1 or TUP1, we get more filaments than the wild type. Again, sticking along with the precedents that can be found in the in vitro literature. However, when we get to the activation of PKA and the PKA function itself, things took a turn for the unexpected. We first looked at the deletion mutant of CYR1, which we had originally got from Jamie Konopka, and we were surprised to see that there's filaments forming here. If we look in vitro, these are round little yeast cells, and so this is a pretty big difference. And since CYR1 is absolutely required for activation of the protein kinase module, we found this fairly unexpected. We then made a TPK1, TPK2 double mutant and asked if that strain can form filaments in vivo. And indeed it can. Whereas in vitro, again, nice, round, well-behaved yeast cells. So this is a fairly significant departure from the standard models that are in the literature about how the protein kinase A pathway functions during filamentation. And this basically says the cyclic AMP pathway, first of all, cannot be the prime regulator of EFG1. And second of all, it can't be involved in the downregulation of NRG1 or TUP1. Something else has to be doing that. And so one of the ways we next looked at NRG1 and TUP1 themselves and asked if they had similar functions in vivo. As we've shown, the NRG1 and TUP1 mutants form increased levels of filaments consistent with their in vitro data. We then asked if this activation or increased filamentation in the mutants of these two repressors was due to or dependent upon EFG1 itself. And again, surprisingly, when we knocked out EFG1 in the NRG1 background, we still had filaments. Whereas when we knock out EFG1 in the TUP1 background, we only form yeast. And so what this means is that NRG1 and TUP1 cannot primarily function as a complex during filamentation. TUP1 does not have an DNA binding domain and is dependent on another factor for its function as a transcriptional regulator. It also is important here to know that EFG1, which in the standard model, NRG1 leaves the promoters of hyphaspecific genes, EFG1 comes in to turn those genes on. Well, if the double mutant still forms a filament, it doesn't seem like those genes are dependent on EFG1. And in fact, when we look at gene expression in these single and double mutants, we see exactly that. So on this graph, on the big, we have green, we have sort of salmon, and we have blue. And those are the expression in the EFG1, ROB1, and BRG1 mutants. And we have this for ALS3, ECE1, HWP1, IHD1, SAP6. All of these are hypha-induced genes. And their expression is reduced in our core regulators. When we knock out NRG1 in the EFG1 mutant, this goes right back to wild type levels of expression, indicating that EFG1 is not required for the activation of ALS3, ECE1, or HWP1 after relief of NRG1 expression, which is somewhat different than what you will read in most reviews of the regulation of hypomorphogenesis. And when we look at all these different genes, we see the same effect until we get to SAP6 and UME6, which is the two genes where we see that EFG1 appears to play at least a partial role in the regulation of those transcripts. And so this, again, a very different model emerges from our studies in vivo compared to what has been revealed by in vitro analysis. And so to finish up here, our new kind of regulatory circuit as it is for in vivo filamentation in the ear model is as follows. We have a cyclic AMP PKA independent circuit in which TUP1 appears to function through EFG1, probably through negative regulation. EFG1 appears to primarily function to relieve NRG1 repression. It is not required for the activation of the hyphaspecific genes. That job seems to be primarily driven by BRG1, and in data I haven't shown you TEC1 as well as ROB1. And so this circuit, again, is slightly different in some fairly important ways from what we have gleaned over the years from in vitro analysis. And so one of the things we're looking into in the future is trying to understand maybe what protein kinase signaling pathways are driving this, and what are the other intricacies that are specific to in vivo filamentation. And so, as I mentioned, Rohan Waqaid has really been done most of the work on this project. Tommy Ollinger, Melanie Wellington, my longtime colleague and collaborator, was the one who first developed the ear model for our imaging, and Uri Kamara helped make the protein kinase mutants that we studied. Scott Filler, Aaron Mitchell, and Jamie Konopka are also collaborators, have authored lots of nice help and advice as we've gone through this project, and our funding is from the NIAID, and I thank you for your attention.
Yeah, thank you very much. And we will now enter the discussion. We will give you a rest, Damian, and start with the discussion for June. Just wait for her. Yeah. there we are. So I'll shoot out some questions and give you time to answer, and then we switch, and then we switch back. And the first question that I have here is actually, if you look at human patients, is there anything known about an effect of antibiotic treatment in early childhood on the development or risk for development of diabetes? I guess that's a question that you thought of yourself.
Yeah, so there is some data in humans to suggest that early life antibiotics has an effect on later development of diabetes. But again, beyond that, there's not much known.
Yeah,
So
and can you say something about what would be the time window in a human newborn or baby? Is that known, or can we give estimates for that?
So before the age of 3 seems to be important,
Mhm.
as far as I know. But again, I don't think like the most perfect studies have been done where they've captured like all of the different developmental windows that you're asking about. So some of this stuff just needs to be captured a little bit better in the literature.
Okay.
It's a
And I
great
have
question.
another question. Sorry, yeah. Probably one of the key questions that has to be addressed after your data as well. So there's another question. You showed a lot about the composition of the microbiome and its impact on the beta cell mass. What about the diversity itself? Does it matter in the same way?
So, well, in our studies, we obviously are decreasing diversity while we antibiotic treat. What we haven't done a really great job of doing is really analyzing the communities that kind of rebound back afterwards. My knee-jerk response is that there are specific organisms that matter at this point in time. So I think that the composition is really important, like the types of microbes that you have. I don't think it's just one, and I think our data supports that there are multiple organisms that are going to have a positive effect on beta cell development. But again, these are all experiments that need to be done.
Okay, and I'll take a final one first for the first round, and this is about the Candida species. So there is a lot of questions on the secret Candida species. Can you give the name and the whole genome sequence and everything? So is there anything else that you can tell us about this species?
Yeah, so this species is found in human infants. I'll give you the name and whole genome sequence when we publish the paper. You know, I'm just because I know that this is recorded, and so I was being a little secretive on purpose. But it's highly related to Candida albicans. It's not a novel species; it's a known species, but you do find it in human infants. It's known to be less virulent, as one could probably guess. But hopefully, this paper will be published soon, and the name of it will be revealed. And we don't know the exact mechanisms by which this organism is doing it. So we're figuring that out now.
Perfect.
I know
I
it's
think
disappointing.
everyone will understand this. Okay, Ilian, over to you.
All right, so the next question is to the main. Somebody is asking if the filamentation circuit is different in a context-dependent manner. So can you speculate, or do you know of environmental factors that allow for this alternate circuit function in vivo and in vitro?
Yeah, no, that's a super good question. Really, you know, the in vitro studies are great at sort of providing one or two or maybe a few of the well-characterized signals that can lead to the formation of this. Probably the simplest answer is you're getting them all at once. And so you're getting a multifactorial set of inputs to drive filamentation, and that leads to different combinations of response pathways. You know, some of the things that we see certainly that could differences between in vitro and in vivo filamentation. We would like to know more about what the expression profile in vivo is and how it compares right in vitro. The data I showed was for a nanostring experiment, which is only 150 or 180 genes. So we're not looking genome-wide. We have just recently been able to do RNA-seq directly on ear tissue and get signal for Candida. And some of the things that we see are that the glycolytic gene expression is very, very different between in vitro Candida and in vivo Candida. And so one of the possibilities is there are pathways that they're responding to different nutrient stresses that aren't being well mimicked in our in vitro conditions. And the other set of genes that goes up a fair bit that was somewhat surprising is the cell wall genes are actually quite different. The expression profile of cell wall genes in vivo is very different from in vitro. And so we're hoping to take this more broad genome-wide look at the expression and try to narrow down to maybe more specific inputs that are sort of being missed in vitro, and we can then maybe make a better model, you know, with a new set of in vitro conditions.
Okay, thank you. In this set of questions, while we are on filamentation, the other question is if the in vitro filamentation percentage changes, but the in vivo, it doesn't change that much in the time course. But filament length changes. So do you have any thoughts about that?
So the filament length stays about the same in vivo or in vitro, even though we start to lose the total percentage of filaments goes down in vitro. And what we're seeing there is that's actually the lateral yeast formation. So in vitro, you see hyphae back to yeast transition. And we see expression of a gene called PES1, which was described initially by Julia Kohler as being critical for that transition. In vivo, we don't see that expression. And we don't see any of the lateral yeast formation. And so the significant difference between these is that the filament back to yeast transition does not seem to be happening in vivo, but it is happening in vitro. And in some data from a paper that's under review right now, if we overdrive PES1 in vivo, we can start to see this lateral yeast formation. It's possible that this happens later. So we're looking at 24 hours. It may come later at later time points, but where we're looking right now, the significant difference is that this the hyphae back to yeast transition is not happening in vivo where it is in vitro.
Yeah, very different environments, very interesting direction. So I'll pass now to Oliver.
Right, June. I'll continue with this question. Is there, so you showed this effect of macrophages also on the beta cells. Is there example of pathogens that encourage or stimulate macrophages or any other immune cells to also attack the beta cells?
I am not aware of this. I saw that question come up after I was giving my talk. I'll have to look that up. I'm not aware of a pathogen attacking a beta cell and stimulating this. There's, of course, in zebrafish, Karen Gilliman's group has studied that there's a commensal organism that makes this antimicrobial molecule that looks very much like the reges that we produce. And this molecule from the microbe can stimulate beta cell proliferation. So there's a precedence for organisms, I guess, kind of not attacking the beta cell, but at least stimulating the beta cell. But I am not aware of like a specific pathogen. So it's something I'll have to look up. It's a good question.
Okay. And then there's another question here, again about the Candida species, but this time you showed that this is related to the macrophages. Is this dependent on pectin-1, or do you know anything about receptors required for this?
Yeah, well, the answer is no, we don't. We have to do those experiments. So I want to point out that we're talking about neonatal mice. So these mice are tiny. And then we're talking not about macrophages in the total pancreas, but macrophages that are present in the islet themselves. So studying these macrophage populations is very difficult because at that time period, there's very few cells that you can get out. We have done a single cell seq on the macrophages isolated from a neonatal islet. And not surprisingly, or actually it was surprising to us, is that the gene signature of a macrophage isolated from an islet of a germ-free mouse versus an SPF mouse is not very different. The major difference is just the sheer number of macrophages that are seeded into the pancreas itself. So whatever the mechanism is, it might be Dectin-1 driven, it could be TLR driven. It seems to be affecting either the development of the macrophage or the seeding of the macrophage population into the pancreas. So we're going to start looking at, early events involved in macrophage development. And that could even be, in the bone marrow. These microbes could be affecting what happens in the bone marrow.
And what then do these macrophages in the islets do? Do they directly stimulate growth of the beta cells, or what is the mechanism there?
No, there's lots of known mechanisms by which macrophages influence beta cells. Of course, I don't know what we're looking at in our system, but the macrophages provide a lot of growth factors to beta cells, whether that be TGF-beta or EGF. They provide a lot of these factors. So they are important in the islet for stimulating proliferation and growth. And they're also important for clearing off any apoptotic beta cells.
Okay, back to you, Ian.
Okay, so the next question is more technical. Is there any actually difference if you vary the inoculum? So you see this linear effect. Are you sure you haven't saturated the air or the media, or do you get a linear response varying the number of cells?
So we have used a single inoculum for these studies. So in terms of saturating the cells, I can't really speak to that. But the inoculums are prepared the exact same way for both in vivo experiment and the in vitro experiment. For instance, for the TPK1, TPK2 experiments, we also checked to make sure that we didn't get a suppressor mutation in that, and took the inoculums and compared them side by side under those conditions. So that, but I can't tell if we've never sort of done a dose response where we look to see if at low inoculum in the ear, we see one rate of change or developmental state versus another. It's all been pretty much at the same one that's mostly been chosen so that we can get good amounts of data for the experiment.
So it's from the same person asking here. I kind of cut the question in two. But have you thought about the quarrel sensing issues in that situation? So do you control like pH, temperature, and so on in the inoculum?
Yeah. So we have not looked at any mutants necessarily that have roles in quorum sensing, although PES1 interacts with farnesol as part of this hyphae back to yeast transition, and the pattern we see doesn't fit the farnesol effects that have been reported in vitro. But that's an interesting question about whether that's limiting, the total. We get to this point and we don't really see. We have a static situation where we have pretty much the same ratio. It's not that every cell turns into a hyphae. And when we look in pathologic samples from patients, you see distributions of yeast and hyphae within that tissue. So we're seeing the same thing. And what's driving that sort of ceiling to the total number of filaments that's being formed? Quorum sensing could be part of it. There could be other factors for sure. It's an interesting question, yeah.
Okay, so I will go with the third question here. So it's more on a cellular level. So in vivo, can you follow basically the single germ tube in 3D as it forms complex branching mycelium in the tissue? It's more if you can distinguish old and new hypho compartments that can actually change in expression pattern.
So in the time course that we're looking at, we see almost no branching. And so we're basically, and we've looked at that and we've measured it. Actually, you can, if you want to look in our eLife manuscript, we characterize that. It's very low numbers of branching. And this is done in confocal. So we take multiple stacks and we follow individual cell bodies down through in 3D in order to differentiate, a hyphae cut through in cross section is going to look round. So we want to make sure when we call something a yeast, it's really a yeast and not just a cross section of a hyphae. And so the lengths also seem to, I mean, we see some that are quite long, but there's a distribution. So I don't, it's certainly the case that our length measurement is a estimation, because we take those, in order to measure the length, it takes way too much computing power to actually follow that all the way down through. So we squish it and do a 2D representation. So absolutely, the length is an estimation, but we do it, these experiments are done wild type and mutant side by side. They're in the same, it's a co-culture. And so it is certainly a relative measure of change in length, but yeah, it's not, it has some characteristics of an estimation based on the data processing we have to do.
Yeah, but then it could, so basically that can lead to some, do you think it can lead to some underestimation of true hypho length? And how do you distinguish branching mycelium from
I mean,
fiber? That's the other part of the question.
So the branching, like I said, we can follow these down, individual ones, and we've done this many, many, many times. We don't see branching. We can see it if we take an NRG1 mutant, actually, has slightly increased branching, and we also have some other mutants that branch, and they make branches that are not very long. So
that's your control in the experiments, basically.
Yeah. And so, in vitro, if you put it on agar, these things can keep making a filament that pretty much goes forever and ever. We don't see that. So the change doesn't, once we get to like 4 to 8 hours, we're seeing the same pattern all the way out. And I'm assuming our error is similar in all of those. So if we're either doing something really systematic to eliminate big hyphae, or we're just not seeing them.
All right, thank you. So we move to Oliver now.
Yeah, June, I have a final question here in the Q&A, and this is if the fungus stimulates accumulation of macrophages in the islets, do you also see evidence of Candida antigen in the islets?
So we have looked to see if Candida is actually in the pancreas, and we have no evidence for that. But I still think we have a few more experiments to do to really rule that out. But I don't think we have any experimental evidence that Candida is there.
Okay.
But that doesn't rule out that molecules could be taken up by macrophages and from Candida and trafficked there. So those are the things we're trying to figure out right now. So it's a great question.
And then I have a question, because so this is probably relating back to my medical studies, where I learned that the development of type 1 diabetes might also be related to viral infections in early childhood. And I was wondering whether you ever considered to also challenge your model with a viral infection in future?
Yes, another component of my lab is interested in commensal viruses. So we've thought about investigating whether or not they're involved in this process. So I think that we will be doing this in the future, but I don't have anyone currently right now. If anyone wants to come postdoc in my lab and answer that question, that would be great.
Yeah, okay. I think you made a good advertisement today. Okay, so Damian, I'll hand over to you, and you have 2 minutes for 4 questions or something. And Ilian.
Okay, so we will go quickly here. So how does the double mutant EFG1 and PR1 is response to pH changes? Have you looked for pH changes in other mutants as well to compare?
We did not look at pH changes specifically with that double mutant. In vivo, what we see is when we look at, so RIM101 is a fairly famous regulator of pH in Canada. It regulates 2 genes, PHR1 and PHR2. One's in alkaline, one's in acidic. If you're in pH 8, it's all PHR1. If you're acidic, it's PHR2. We see a mix. And when we look at the effect of that distribution and compare it to varying the pH in vitro, the pH seems to be about 7.1, 7.2-ish, if you want to use that. So we don't have, there's lower expression of the acidic component, but it's not 0. And so this is not, so that's the best we can do with that. And RIM101 is having an effect. It's one of the sort of auxiliary regulators. However, if you take a RIM101 mutant and put it at pH 7.5, it won't make any filaments at all. And we see some filaments in the RIM101 mutant. So it has to be somewhere between 7 and 7.5 in vivo.
Okay, thank you. And another more practical question, what in vitro phenotypic assay for filamentation matches best the in vivo assay in the year? Can you recommend something?
Yeah. Probably RPMI plus 10% serum at 37 gives us a pretty consistent, reasonably consistent. Where we see the most deviation is on plate-based assays. And so most of the transcription factors that were only showing phenotypes in vitro were based on plate-based assays. But that and $2.50 will get you a cup of coffee at Starbucks. I wouldn't hang my hat on that analysis very much. That's the best we can do. So maybe one day I'll, do a Harry Potter Professor Snape thing and just start throwing stuff together and see if we can't make it a match in vivo. We'll see.
All right, thank you. And there is an Outlook question more like, is crypto next?
No.
No, okay. That was a short one. Okay. So with that, Oliver?
Yeah, thanks. So first of all, apologies once again to both speakers for the chairs somehow dropping out due to technical issues. But you basically saved the session together with Gordon, I should admit. It was really cool to listen to your talks. Thanks to everyone for participating. And remember that the next MycoTalks will be on 29th of February. Thank you very much and have a nice evening.
Thanks, everyone.
Thank you.